Sobrang tagal na boy. Good morning sir, good morning classmate. My title report for today is Trends in DNA Technology. Introduction. To study this unit, you should be first understand the concept called DNA. The DNA deoxyribonucleic acid is the molecules of inheritance with hereditary information are stored in virtually every organism. It is located in the nucleus of cells found on the body. This is titled This is Little Knowledge of DNA, while assist when you going through the lesson as we would. Trends in the development recombinant DNA technology. Our knowledge of living cells date to 1665 when Robert Hooke described the space. He observed a slice of cork in 1838 and 1939. Snyder and Schumann formulate the cell theory, show that the cells is the basic organism unit of the living organism. On the closer look, one should be appreciate the fact that the living cells is the epitome of indigenous design. All through the DNA was the first isolate 1869 by Swiss biologist Frederick Mischer. It was not unit 1943 that DNA was conclusively identified as the primary genetic material. The brilliant experiment that was performed MacLeod and McCarthy, who invested the previous finding Griffith. From the point of the space of the research of DNA, molecules become rapid and intense. The foundation for what is known now as recombinant DNA technology of genetic engineering was laid by Watson and Crick. 1953, a British journal, Nature, that came up with the finding on the double helical structure of DNA and the purpose of mechanism of DNA replication. After then Comberry proposed the mechanism of DNA reflection. After then Comberry 1958 isolated DNA polymerase. Between 1961 and 1966, the genetic code was a complete workout. In 1963, the discovery of the reverse transcriptase called regarded as another landmark of recombinant DNA. The case in 1967 and restriction enzymes in 1970. The stage was set from the take-off of the new technology, genetic engineering, armed with these three types of enzymes. Reverse replication DNA, the case of restriction, endonuclease. Scientists now have the ability to create new form of life by splicing together DNA. or genes from virus source. In fact, the first recombinant DNA molecules were systemized at Stanford University, USA in 1972. The following year, the Pandora's box was opened at the Gordon Conference on Nucleic Acid held in New Hampshire, USA. Thank you, Sarah.